This is the story of Rickettsia. You may have heard of its reign of destruction, even if you haven't heard of it. To microbiologists, Rickettsia is enigmatic. And while she may not be taking the world center stage yet, she may soon, after global temperatures start to rise. This teensy microorganism has hikers shaking in their boots, mostly for its link to the tick-borne illness Rocky Mountain spotted fever, but also for its role in typhus in the days of yore. She's a gram-negative, pleomorphic, obligate intercellular parasite, an itty-bitty, just 0.2 to 0.4 micrometers long on average, allowing her to thrive in intercellular spaces. Her adhesion proteins allow her to stick to cell walls, where she can linger before being ingested. She's got a wild ability to recombine her DNA, fortifying herself with DNA from other organisms and antibiotics, making her highly evolved and difficult to kill. She's an alpha protobacteria in the Rickettsia family. She's got 25 catalog species in her genus, depending on the immune response that they trigger, typhus or spotted fevers, but also many non-pathogenic subgroups. Most types of Rickettsia only infect insects, fleas, ticks, chiggers, or aphids, but when they migrate into vertebra, they can cause a cellular ruckus. Poor Rickettsia, though, she's wholly dependent on her host to survive. She uses a unique protein that enables her to import ATP directly from her host because she's unable to use metabolites on her own. She's particularly destructive. First, she clings to her cell wall with adhesions, and then she escapes the vacuole after entering the cell, propagating through binary fission, eventually creating a necrotic cell that bursts within a day or faster in weak immune systems. Many species of Rickettsia, though, are innocuous to humans. They live in a happy symbiosis with their arthropod hosts. However, there are several types that are harmful to humans and deadly. Blood-sucking insects feed on infected hosts, like mice, deer, livestock, and hikers, where the bacteria can enter the host body. If they remain on a human undetected for over 24 hours, the likelihood of vector transmission into the human is high. After being bitten, the damaged vascular tissue will leak blood, causing what looks like a rash from an infected host skin, hence spotted fevers. Eventually, this ruined tissue will lead to strokes, renal failure, vascular problems, and eventually death. If it's undetected, within a few weeks of infection, Rickettsia can be treated with antibiotics. But higher global temperatures have led to longer tick seasons and new regions for infected arthropods to survive, such as hiking trails in New England. Particularly, these areas have exploded with outbreaks. So hikers beware. This is Rickettsia, and she is coming.
Hello everyone! Today I'm going to be talking about Chlamydia trachomatis. Chlamydia trachomatis is a gram-negative, obligate intracellular bacterium that targets the reproductive tract through mucous membranes in humans and is the #1 most common sexually transmitted disease in the world. Studies show that over 131 million cases of Chlamydia trachomatis microbial infections occur annually. The shape and anatomy of Chlamydia trachomatis consists of two phases of development. The elementary body, which is the infectious extracellular form with a clear bubble-like wall in its first phase, which then becomes internalized within the endocytic vacuoles and transforms into its secondary body called the reticulate body. The reticulate body splits itself into two identical coccus-shaped copies, which then become capable of stealing nutrients from the host. Once full lysing of bacteria occurs before transforming back to its original elementary body and continuing infection. This lifestyle of switching back and forth between body forms is its strategy for survival. Chlamydia trachomatis is closely related to 3 other species amongst the Chlamydia genus. The others include Chlamydia pneumoniae, Chlamydia psittaci, and Chlamydia pecorum, which all similarly cause human respiratory infection. Chlamydia trachomatis is commonly thought of as a virus because of its dependency on the host, but because it has a cell wall with DNA, RNA, and ribosomes, it is classified as a bacteria. During the stage of infection, the elementary body, which is metabolically inactive, enters and binds to host cells by way of endocytosis. When arriving in the genital tract, cells change form in their reticulate body and attach so the bacterium can evolve by gathering nutrients like ATP, amino acids, and nucleotides synthesized by the host. After consuming these nutrients, a moment of high metabolic activity is engaged, and the power of developing more DNA and RNA accumulates inside the inclusive outer layer. The inner reticulate body replicates several times by binary fission before lysing more bacteria and returning to its elementary body. Chlamydia trachomatis will interact with a variety of different microorganisms in the genital and gastrointestinal regions, the most being Neisseria gonorrhea. Together they join forces to amplify each other's growth and cause more pathogenic harm to the target area. Microbes associated with bacterial vaginosis can interact with Chlamydia trachomatis and worsen symptoms due to unbalanced pH levels, inhibiting the mucosal security within the vagina. HIV interaction often dominates the body's ability to fight off Chlamydia trachomatis, which forces engagement and weakens immunity. Evolutionarily, the Chlamydia genus shares similar traits and features to other intracellular bacteria. The ancestors of Chlamydia trachomatis were likely free-living in open air or surviving underwater, eventually adjusting to be able to survive in host cells. This adaptation caused genome reduction and the loss of independent metabolism, which shifted its existence from harmless to pathogenic. Chlamydia trachomatis spreads most commonly through sexual intercourse, anal sex, or oral sex, and is often referred to as the silent infection because of its asymptomatic tendencies. Symptoms present will be experienced differently depending on the female or male anatomy. In females, signs involve white or yellow discharge, pain or burning when urinating, itching inside the vagina, pain in the lower abdomen, and bleeding in between periods. However, in male bodies, the bacteria attacks the urethra, causing mucus-like or watery discharge from the penis and burning sensations when urinating. Other than the reproductive tract, chlamydia can appear in the eyes, throat, and anus with similar symptoms of discharge and pain. Chlamydia trachomatis can be treated and cured by taking either a single dose or 1-week round of antibiotics, but if left untreated, can cause serious health risks like pelvic inflammatory disease, epididymitis, pregnancy complications, and infertility. To avoid coming in contact with Chlamydia trachomatis, it is highly encouraged to abstain from any sexual activity and fluid bonding with anyone who is infected. Preventative actions can include using condoms and regularly getting STI screenings, especially when symptoms appear.
Hello, everyone, and welcome to my presentation on Lactobacillus acidophilus, a fascinating and beneficial microorganism that plays an important role in both our bodies and the environment. Today, I'm going to be going over my poster as part of the art installation piece of this project. So let's go ahead and start with the introduction. Lactobacillus acidophilus is a rod-shaped, gram-positive bacterium that's widely known for its role in fermentation. It's a major component of the gut microbiome, where it contributes to digestive health and immune defense. You'll also find it in probiotic supplements and fermented foods like yogurt, kimchi, and kefir, where it helps preserve food and enhance flavors. Now let's go ahead and take a look at the anatomy and structure. Lactobacillus acidophilus is a rod-shaped gram-positive bacterium that is non-flagellated, meaning it doesn't have structures for movement, and it's non-spore forming. It has a thick peptidoglycan wall, which is a characteristic of gram-positive bacterium. This structure provides protection and helps it survive in the slightly acidic conditions of the human gut. Phylogenetically, Lactobacillus acidophilus belongs to the domain Bacterium within the phylum Firmicutes and class Bacilli. It's part of the order Lactobacillus and the family Lactobacilliaceae. Its genus is Lactobacillus. This group includes many other probiotic species, such as Lactobacillus rhamnosus and Lactobacillus casei, which also contribute to human health and fermented food production. Moving on to metabolism, nutrition, and growth. Lactobacillus acidophilus is a facultative anaerobe, meaning it can survive with or without oxygen. It primarily ferments carbohydrates to produce lactic acid. This process, called lactic acid fermentation, not only helps preserve foods, but also lowers the pH in its environments, creating conditions that inhibit the growth of harmful bacteria. In terms of growth and nutrition, Lactobacillus acidophilus feeds on carbohydrates like lactose and glucose. Its optimal growth temperature is around 37 degrees Celsius or 98 degrees Fahrenheit, which is close to the human body temperature, and it thrives in slightly acidic environments, just like the human gut. Lactobacillus acidophilus doesn't live in isolation. It interacts with many other microbes. It competes with harmful bacterium such as Clostridium difficile and E. coli by producing substances like bacterioricins and lactic acid. It can inhibit the growth of these competitors. It also forms biofilms with other beneficial microbes, strengthening the overall health of the gut microbiome. The bacterium also interacts with larger organisms, including us. In humans, L. acidophilus enhances gut flora balance and immune defense. Environmentally, it's used in bioremediation and dairy fermentation, particularly in yogurt production. Its ability to modify its surroundings makes it a valuable tool in food science and environmental management. Ending with a summary of its impacts, symbiotically, L. acidophilus is a true friend to our digestive systems. It supports nutrient absorption and helps maintain a healthy microbiome balance. Pathologically, it's mostly safe, but it has been associated with rare cases of bacterium in immunocompromised individuals. And finally, environmentally, it plays a positive role in sustainable food production and waste reduction. In conclusion, Lactobacillus acidophilus is a small but mighty bacterium that benefits humans, other microbes, and the environment in powerful ways. Thank you for watching this presentation, and here are some resources that help me with some of this research.
Hey everyone, so for this project I chose to focus on Neisseria meningitidis. It's also known as meningococcus. It's one of those bacteria that most people don't really think about, but it's actually super dangerous. What makes it really interesting to me is that it can live quietly in our body without causing problems, but when it turns into pathogenic, it can become deadly really fast. And I've experienced that firsthand because my friend actually got meningitis, and it was kind of rare coincidence. But also working in the hospital, I have seen it firsthand and how fast this micro can change someone's life, and that's what made me really want to learn more about it. So I will introduce you to our guest today. What's up everyone, my name is Neisseria meningitidis, but you can call me Meningo. I'm one of those bacteria that you don't really notice until it's too late. Most of the time I'm just chilling in your throat, no issues, no drama, till things get dramatic, and that's when I get really spicy. As you have seen, under the microscope, I look like two little coffee beans chilling together. That's something you guys called Diplococcus. I'm a gram negative. I have a special outer membrane that stains pink in the lab. I don't have legs or flagella, but I do have something called pili. Think of them like a sticky tentacles that help me hold onto your throat. My capsule is very unique. Usually it's my hoodie. It's very slimy and thick, and it keeps your immune system from touching me. It also decides which squad I belong to, because I could be in A squad, B, C, W, Y, or X squad. Some of us are more dangerous than others. Vaccine try to keep up with us, but not all of us are covered. In order for me to grow in the lab, I'm a bit picky. I need a chocolate agar, not the actual chocolate that you guys eat, just some extra rich media to grow on. I'm aerobic, I use glucose and maltose for energy, and I'm oxidase and catalase positive, and that's how scientists can reveal my identity, because I'm bougie like that. And honestly, you don't gotta do much to spread me. A cough, a kiss, or even sharing drinks, that's all it really takes. I love packed places like college, dorms, classrooms, parties. And the twist about me is that most people carry me and they don't even really get sick. And they pass me around like I'm a group project. But when the opportunity comes, that's when I get you sick. I make my moves, I sneak past your defenses, enter your bloodstreams, maybe even hit your brain. That's how spicy I can get. Anyways, just to let you guys know, I'm not alone in this. I have got family in low places too. My cousin Neisseria gonorrhoeae causes gonorrhea, which is a really nasty STD. We both come from the same family, Neisseriaceae, but I'm picky. I love to live in humans, no animals, no dirt, just people. And according to your guys' CDC, around 1 in 10 people carry me in their nose or throat, and y'all don't even know it. But unlike my cousin, I don't mess with your private parts. I go straight to your brain and your blood. I cause something called meningitis. Hence, they call me meningo. That's something that causes an inflammation of your meninges, the protective layers around your brain and your spinal cord. Some of the symptoms I cause include fever, headache, and I love giving you a stiff neck. And if I really get going, oh boy, I will make your blood vessels dilate, drop your blood pressure, start to shut down your organs, and if I really feel like taking you on a crazy ride, I will cause something called sepsis. That's something you don't want to mess with because it's a whole bloodstream infection, and pretty much I put your body into a war zone. And here's the wild part. I don't need days to cause all this mess. I can take someone from I feel fine to ICU admission in like 12 hours or less. Blink and I'm already in your spinal fluid. But hey, humans have been getting smarter. You guys have gotten vaccines now for type A, C, Y, and W, and even type B, but guess what? We always adapt and we will always evolve. Type B can be a little tricky because guess what? I can change up my capsule like an outfit. And that's something called genetic recombination. It keeps your immune system from guessing, and it makes vaccines a little trickier. And if you attack me too hard with your immune system, guess what? Boom, I have another plan for you. And guess what? Even if your immune system was able to take me down, well, you're going down with me too, because I dropped something called LOS, which is a lipo-oligosaccharide. You probably haven't heard of it, but it's a toxin bomb. It causes so much inflammation that your own body ends up doing the damage itself. It's like I throw the match and your immune system burns the whole house down. I've gone a little dramatic, but anyways, doctors can usually spot me with a spinal tap or a blood culture, but they don't usually wait. As soon as they suspect I'm in the house, they drop those good antibiotics. Because time matters, because guess what? If I settle in, it's a wrap for you. So yeah, I might be small, but I'm no joke. I'm Neisseria meningitidis. I'm silent, smart, and deadly. But if you stay vaccinated, you stay clean, and you don't share your smoothies, you'll probably be fine. Until next time, peace out.
Hello, everyone. Today I'll be talking about an important group of microorganisms that live on and within us, the Staphylococcus genus. Staphylococcus is one of the most common types of bacteria found on the skin and mucous membranes. Of all the species in this genus, Staphylococcus aureus is the most well known. Under the microscope, all of these bacteria are in grape-like clusters, which is one of their most distinguishing features. They are classified as gram-positive bacteria because they have a thick peptidoglycan layer in their cell wall. Most species also have a polysaccharide capsule, which actually helps them evade detection by the immune system. Additionally, Staphylococcus bacteria are non-motile, non-spore-forming, catalase positive, and capable of living in both oxygen-rich and oxygen-poor environments. From a classification standpoint, Staphylococcus belongs to the phylum Firmicutes and the family Staphylococcae. Apologize if I pronounce those wrong. One of the most remarkable features of this genus is its metabolic flexibility. While glucose is their preferred energy source, Staphylococcus species are heterotrophic, meaning they can also metabolize other organic molecules. They primarily generate energy through glycolysis and reproduce through binary fission, which, as we all know, means dividing into two identical daughter cells. When it comes to interactions with other microbes, Staphylococcus is competitive. It fights for nutrients in its environment, but also forms biofilms with other bacteria. These biofilms can enhance survival and facilitate horizontal gene transfer, which, as we learned earlier in the term, can aid in spreading antibiotic resistance. Most Staphylococcus species are harmless and even beneficial, particularly on the skin. However, some, like Staphylococcus aureus, can act as opportunistic pathogens. S. aureus is responsible for a variety of infections, including skin abscesses, pneumonia, endocarditis, toxic shock syndrome, and even MRSA. MRSA is a global health concern because of its resistance to multiple antibiotics and its role as a leading cause of death due to antimicrobial resistance. Though not all Staphylococcus species are the same. For example, Staphylococcus epidermidis actually plays a protective role. It helps maintain the skin barrier, support immune function, and even contributes to wound healing. So, in conclusion, the Staphylococcus genus includes some of the most important bacteria living on the human skin. While certain species can cause serious illness, others are essential for maintaining a healthy skin microbiome. Understanding this balance is key to both preventing infections and promoting overall skin health. Thank you so much for listening. I hope you learned a little bit more about the Staphylococcus genus. And here are some sources that I used in order to find all my information and get a better understanding of this. Have a great rest of your day. Bye-bye.
E. coli is the most studied and best understood bacteria to date. There are nearly 1,000 known strains of E. coli, with new ones still being identified. Commensal E. coli lives naturally in our GI tract, aiding us by taking up space that could otherwise be taken up by more pathogenic microbes and even other strains of E. coli. But even this commensal E. coli is capable of causing UTIs, sepsis, meningitis, and various enteric diseases, as it is an opportunistic pathogen. Other strains have varying virulence factors. E. coli is a gram-negative. It does not produce endospores. E. coli is rod-shaped and appears solo or in pairs. It may be motile with the presence of pernacious flagella. It is a facultative anaerobe. E. coli belongs to the Enterobacteriaceae family, like Shigella, Salmonella, and even Yersinia pestis, which cause the plague. It is acid tolerant and can survive in the pH of stomach acid for hours on the way to the intestines. E. coli is found in the mucus and epithelial cells of the intestines and make up to 1% of gut bacteria. Metabolic requirements are flexible, as E. coli has the ability to switch pathways when nutrient availability changes. Pathways such as glycolytic pentose phosphate, TCA cycle, and fermentation are some of the pathways. The source of nutrients of commensal E. coli in the biofilm are shed epithelial cells, dietary fiber, and mucosal polysaccharides, but each pathotype has specific nutrient needs based on the host microenvironment. E. coli can conjugate with other microorganisms, exchanging genes and ever evolving. This is done by extending appendage called pili, making contact with another bacterial cell and transferring genetic information. But the most frequent way that E. coli exchanges genes is by bacteriophages, viruses that infect bacteria specifically. Some of the pathotypes of E. coli are enterohemorrhagic E. coli, which produces a Shiga toxin. It is able to survive through innate host defenses like saliva and stomach acid because of its acid resistance. Once in the intestines, the Shiga toxin causes chaos, resulting in bloody diarrhea, renal failure, dehydration, fever, and fatigue. Enteropathic E. coli doesn't produce toxins, but it does attach interface microvillus. It causes persistent diarrhea and is a leading cause of death of infants in developing countries. Enterotoxigenic E. coli causes diarrhea by secretions of enterotoxins. Enteroinvasive E. coli is similar in many ways to Shigella, and this strain causes severe watery diarrhea, fever, and cramps. It may be local in the intestines or systemic. E. coli is found in the feces of animals, and the transmission of bacteria may begin with fecal matter contaminating food directly or contaminating water. Contamination that reaches water may be used in agricultural irrigation or reach waterways that lead to recreational waters and beaches. The USDA and FDA have guidelines to reduce transmission, which include inspecting facilities and testing food. The most common food contaminated are fresh produce, raw dairy, and undercooked beef. But it's not all so bad. E. coli has been the model of choice in the laboratory since the 1940s. And microbiology, as we know it, wouldn't be here today without E. coli.
Hello, I did my term project on the cellulitis species. So a little overview of what I'm going to be talking about today is we're going to explore the impacts of the cellulitis species along with its shape, anatomy, classification, what it is, its metabolism, growth and development, and nutrition requirements, along with its interactions with other microbes and its environmental impacts. So my microbes overview, what is a cellulitis species? It's a gram-positive bacteria that's associated with infections. It can be found in soft tissues and skin infections, and it contributes to the skin's redness, inflammation, pain, hotness, and tissue damage. This can be found anywhere, but it's most commonly found in people's legs. Why it's important, it's has a potential to cause major infections if it's not properly taken care of and treated early on. So it's important to be aware of this so you know how to treat it and how to identify it. So its host interaction, the bacteria enters through cuts, wounds, abrasion, burns, and bites, so any opening on the skin. And then it maneuvers through the dermis layer of your skin, but it can also go deeper than that. It triggers immune responses, creating the swelling and redness. And the deeper it goes, the more serious the condition. There's a sepsis condition that is known to be kind of like permanent, so it can be very bad. Favorable environments, it survives and reproduces best in moist, warm, and damp environments. And it thrives in dirty sections. So if somebody doesn't have the best hygiene, then they're more likely to get the cellulitis infection. Its shape and anatomy, it's a gram-positive cocci. It's a grape-like clustered for the Staphylococcus aureus. And it's non-movable, like it can't move around without flagella, so it needs that flagella to give it its movement. Its anatomy and features, it's non-spore forming, and it has a thick peptidoglycan cell wall, and it makes toxins and enzymes that help invade and infect our tissue. Its classification, it belongs to the Staphylococcus in Staphylococcus aureus species, and it can be classified based on the 165S rRNA sequence. So what are its nutrition, growth, and metabolism needs? So as far as nutrition, its main energy source comes from glucose, and it needs specific amino acids, minerals, and vitamins from us, the host. And it grows best in conditions that are anaerobic fermenting. So places with low oxygen or just not much oxygen present. And its favorable temperature is 37 degrees Celsius, which is actually our body's normal temperature. So that makes us a favorable environment, hence why it clings to us. As far as metabolism, it ferments glucose and then it produces lactic acid as a byproduct. Interaction with other microbes. So it's actually competing against the microbes on our skin that help us function normally. It's trying to take over those and take away the machinery and invade that area. So it's a lot harder on our body to be able to fight back against the infection when those are being taken over. So it harms the balance of our body's natural microbiome. So overgrowth of the cellulitis can lead to an infections when the skin's natural defenses are impacted and then they lose control. So health impacts, normally it's a pretty mild infection if it's treated properly and early on. So you won't have long-term symptoms as long as you're on top of it. But if you don't take care of it, it can lead to you needing amputation, and it can lead to just permanent tissue damage that you'll have to live with. This is more commonly dangerous and found in people that have diabetes, cancer, HIV, or a little bit more overweight. They become more susceptible to it, and it's harder for their body to fight it off as easy because of their weaker immune system. So when it's in the favorable environment, such as those people, kind of takes over for them a lot quicker and it's more severe. So in conclusion, the cellulitis species is an important pathogen in creating soft tissue damage and infections. Its interaction between other microbes and hosts can lead to a more severe infection. The better we're able to understand the anatomy and its effect, the better we'll be able to treat it early on. And the quicker you're able to diagnose the pathogen, the less likely you're to have long-term effects such as tissue damage or sepsis. And here is my bibliography if you guys would like to dig into it deeper. Thank you.
Hello, and welcome to my presentation on my favorite microbe, Rhizobium. Part of why Rhizobium is my favorite microbe is because of its importance within our environment. I'd like for us to first look at its main function within the biosphere, and then speak to its characteristics and anatomy that allow the various species within the Rhizobium genus to carry out their amazing work. There is a reservoir of nitrogen gas within our atmosphere, but it is relatively unusable. Some microorganisms have evolved to convert this gas into a usable form via biological nitrogen fixation, and Rhizobium is but one genus that has this capability. This process of nitrogen fixation is called diazotrophy, and is unique to prokaryotes and is thought to be the result of coevolution with flowering plants. This biological process of nitrogen fixation allows for atmospheric nitrogen gas to be converted into ammonia, which is essential for growth and development of plant life. In the case of Rhizobium, there is a symbiotic exchange of molecules between the host plant and the bacteria. The plant obtains usable nitrogen in the form of ammonia, and the Rhizobium obtain carbohydrates and other minerals necessary for continued metabolism from the host, along with protection from the environment by the formation of root nodules. Many other genera of bacteria are also able to symbiotically and biologically fix nitrogen, such as Azotobacter and Frankia, but Rhizobium is specifically known for its symbiotic interactions with plants in the Leguminosae family. Now that we have a general idea of why nitrogen fixation is crucial to our ecosystem, let's now take a look at the specific structures that make Rhizobium capable of this biological process. Rhizobium species are gram-negative, rod-shaped bacillus bacteria. As they are gram-negative, they have a cell wall with two membranes, an inner thin peptidoglycan layer surrounded by an outer lipid layer composed of lipopolysaccharide proteins, allowing them to be more resilient to environmental stresses. They are motile, possessing flagella, which are particularly important for early stages of root colonization as it allows for interaction between root hairs of the host plant and the bacteria. Rhizobium also have a polysaccharide capsule, which aids in attachment to root hairs and protection against desiccation. A particularly fascinating anatomical shift occurs, however, when the bacteria are encapsulated by the root nodule. When in the low oxygen environment of the nodule, they differentiate into bacterioids, irregular or Y-shaped cells adapted for nitrogen fixation. The low oxygen environment is essential for the functioning of their nitrogenase enzyme complex, which allows them to fix atmospheric nitrogen and convert it into the usable form of ammonia. Phylogenetically, Rhizobium belong to the alpha subdivision of Proteobacteria, which includes phototropic genera, several genera that metabolize C1 compounds, and bacteria that are symbionts of plants. They are part of the order Hyphomicrobiales, an order of gram-negative, non-spore-forming, rod-shaped bacteria, and they are from the family Rhizobiaceae, which is symbiotically associated with legumes. The family Rhizobiaceae is also classified into 6 genera. The fast-growing genera Rhizobium, Allorhizobium, Mesorhizobium, and Ensifer, formerly Cynorhizobium, the slow-growing genus Bradyrhizobium, and the genus Azorhizobium. 2 common species within the Rhizobium genus are Rhizobium leguminsarum, which is known for its association with peas, lentils, and other legumes, and Rhizobium tropici, which is commonly found in root nodules of tropical legumes. There is a large amount of genetic variation even within these species due to the large mobile plasmids, known as symbiotic plasmids or PSYM, that encode genes for root nodulation and nitrogen fixation. The PSYM plasmid, fun fact, is being studied in its ability to be added to the genome of multiple crop species, which would assist plants like corn, which need a high quantity of nitrogen for robust growth, in forming symbiotic relationships with rhizobia in the soil. When not linked to a host plant, rhizobium tend to feed on decaying organic matter within the soil. But how does the nodule form, you may be wondering? The nod factors released by the bacteria are sensed by the root hairs of the legumes. The root hairs, as a result, become more permeable and curl around the bacteria and allow the rhizobia to proliferate around the hairs and eventually infect the plant. As the microbes enter the root cortex, a tumor forms around the colony, and the differentiation into bacterioids occurs. This tumor is what we call the nodule. Another change within the nodule is the production of like hemoglobin, which is able to bind oxygen and thereby reduce the levels of free oxygen, which is a poison to the nitrogen fixation process. Within the soil, rhizobia must compete with other microorganisms as well. In agricultural practices, legumes generally rely on dual symbioses to rhizobia and arbuscular mycorrhiza, leading to less reliance on commercial fertilizers and an increase in sustainable farming. Between the soil fungi and the rhizobial colonies, the combined action assists the host plant in nitrogen fixation. They do compete slightly for nutrients from the plant and the soil, but their relationship is generally mutually beneficial. There is also competition between species of Rhizobium. Rhizobium leguminsarum is less competitive and has a narrower range of host plants, whereas Rhizobium tropici is generally more competitive and has a larger array of host plants. Other nitrogen-fixing bacteria, such as Azotobacter, also exist within the soil, but do not symbiotically link to a host and are instead considered free-living. All in all, Rhizobium plays a vital role in the nitrogen cycle through its symbiotic relationship with legumes, enabling the conversion of unusable atmospheric nitrogen into a usable form for its plant hosts. This genus exemplifies the complexity and significance of microbial association in maintaining ecosystem health and supporting sustainable food production. Thank you for listening to my presentation on my favorite microbe, Rhizobia.
That's how they live. The anaerobes need no oxygen. Oh, actinobacteria related to propion and cornate bacterium. Fused for germs, all your information right. They line together, keep the bad bugs out. Filters made of bifidobacterium. This long gone brevis. The panties are just some of them. Each one has a job inside of you. We sell them packaging so cool. Help with stress and IBS. I say filters made of bifidobacterium. Your futures made of bifidobacterium. Futures made of bifidobacterium.
Hello, I will be talking about Akkermansia, which is a genus of probiotics that are specifically found in mammals. I will specifically be looking at Akkermansia muciniphylla. So Akkermansia muciniphylla is a strictly anaerobic bacteria that is oval-shaped with pili all over its membrane. It is non-motile, gram-negative, and it does not produce endospores. So Akkermansia muciniphylla is a part of the greater PVC superphyla, and then it is a part of the Verrucomicrobiota phylum, Verrucomicrobiae class, the Verrucomicrobialis order, and then the Akkermansia genus. Some other genera found in this phylum are Verrucomicrobium, Prostheticobacter, Luteolibacter, Rosebacillus, Persier habitus, and Rubritalia. Akkermansia is, interestingly, the only member of this entire phylum that has been found in the mammal microbiome, which I thought was pretty interesting. The other genera that I mentioned previously are found in soil and marine environments, and even some of them have a symbiotic relationship with some sponges, some marine sponges. So Akkermansia muciniphylla, mucin within the human gut is the sole carbon, energy, and nitrogen source. And how it metabolizes this is it cleaves the sulfate groups of mucin glycoproteins, and that can be attached at the end of glycan strains. And it also degrades the mucin into acetate and propionate. So why should we care about this? We should care about this because all of those actions that I just previously mentioned are very important to keeping our gut healthy. So when mucin is broken down, the product is acetate and propionate, which I just mentioned, and those are used as substrates for other beneficial bacteria within the gut, which is very important for maintaining the diversity of probiotics within our microbiome. This also produces acetic acid and propionic acid, and this causes a change in the pH of our stomachs, which has shown to kind of increase an anti-inflammatory response within our human gut, which keeps everything balanced, uninflamed, and feeling good. A muciniphylla also reduces endotoxemia by inhibiting translocation of lipopolysaccharide from the intestine into circulation. So preventing those lipopolysaccharides from going into the rest of our body, and just helps get rid of any kind of toxins that might be released through digestion. What if I don't have enough Akkermansia muciniphylla? If you don't have enough of this important bacteria, there's a high likelihood that you probably have a lowered immune response, digestive issues such as inflammatory bowel disease, or a chronic illness such as Crohn's, colitis, diabetes, and other diseases. They have found that people with these sorts of diseases or chronic illnesses have had a significantly smaller colony of a Mucinophylla within their microbiota. 1 to 4% of our entire gut microbiota should be a Mucinophylla. Eating more vegetables can help give our colony what they want, and a lower processed fat diet can improve your gut as well, which are good general rules to go by. Studies are still being conducted on the efficacy of Akkermansia supplements for all individuals, even though we know that the presence of this bacteria is beneficial. They are still doing studies on how to best take it and who should be taking it. But there is a lot of concrete evidence that says that people who have more of this specific bacteria in their system are healthier. So eat some more vegetables, and I hope you enjoyed learning about Akkermansia mucinophylla. Here is the We're Excited. Thank you very much.
Today we're going to be talking about Borrelia burgdorferi. That is the bacterium that causes Lyme disease. So look at this picture. Can you spot the tick? I can't. It can be very difficult to spot a tick on your body. This is our vector, Ixodes pacificus. It's only 1/8 of an inch, and it's located in the far western United States. And it's a vector of Borrelia burgdorferi, or B. burgdorferi for the rest of this presentation. Ticks generally live a highly parasitic lifestyle and rely on hosts for essential nutrients, so they've evolved over time to scavenge amino acids and lipids directly from reservoir hosts rather than synthesizing it themselves. These ticks have a 3-year life cycle. They have a blood meal at every stage of that cycle, feeding on mammals, birds, reptiles, amphibians. They're uninfected when they hatch and acquire B. burgdorferi by feeding on infected reservoir hosts. Once they're infected, they can transmit that pathogen to new reservoir hosts during future feedings. B. burgdorferi is from domain bacteria, phylum Spirochaeta, genus Borrelia. Its relative is Treponema pallidum, which causes syphilis. They're spirochete in shape, long helical corkscrew. They're microaerophilic, which means they need oxygen to live, but too much can be fatal. They do have a peptoglycan layer. They're gram-negative, but have no lipopolysaccharides. They have periplasmic flagella near each end of their body, beneath the outer membrane to help them move, linear chromosomes, and, uniquely, linear and circular plasmids. Some evolutionary strategies that make this bacterium so persistent is that once ticks are infected, spirochetes increase in number and undergo phenotypic changes, including the expression of outer surface protein C, which allows them to invade the host tick's salivary glands, making it easier for them to pass it on to other reservoir hosts. Humans, like my adorable daughter when she was little, are known as dead-end hosts. Humans are not necessary for B. burgdorferi to continue. Accidental encounters between humans and ticks in the natural environment is to blame for contraction of Lyme disease. The CDC receives approximately 30,000 to 40,000 reported cases of Lyme disease annually in the United States, but new estimates show that 476,000 people may be diagnosed and treated for Lyme disease each year. This suggests that there's a significant number of cases that are unreported and undiagnosed. Some signs and symptoms to look for include erythema migrans. This is a bullseye rash around the site of the bite. It usually shows up 2 weeks or sometimes as many as 32 days post-bite. Some patients never even see this rash. They may have fatigue, headache, myalgia, a fever, but they don't see this, which makes it hard to diagnose. There are 3 stages of disease: localized, which you can treat with an antibiotic and have quick resolution.
But then in early dissemination, the bacteria spreads to the bloodstream to other parts of the body. And this is where we see neurological symptoms like facial palsy, meningitis, and even cardiac issues known as Lyme carditis. If you make it far enough in the process, you may contract post-treatment Lyme disease syndrome, which is basically a lifetime of an overactive immune system. So tissue or nerve damage from the original infection persists as inflammation and autoimmune responses. So if you get a tick bite, what should you do? If you're fortunate enough to spot the tick on your body, removing a tick is simple. Clean the area, clean a pair of needle nose tweezers, and use even pressure to pull the tick out, making sure not to twist or leave any body parts, especially mouth parts, in the site. You're going to want to flush the tick down the toilet or put it in a bag for identification. Some further care and treatments. For the first 30 days, it's important to keep an eye out for symptoms, including fever, chills, fatigue, headache, muscle or joint pain. If any of these symptoms should present, see your healthcare provider. And if you find a tick on your skin 36 hours or more after possible exposure, a doctor should be seen immediately, because this is the time frame outside of the B-Burgdorferi migration period. So medical guidelines recommend a single dose of antibiotic, and further treatment may be needed. So see your doctor immediately. If you need more information, the CDC website is fantastic. Go there to find out more about ticks in your part of the United States, as well as things to look out for and treatment plans.
This is Treponema pallidum, a gram-negative motile spirochete bacteria with endoflagella. It causes syphilis, a disease that has impacted global health and history for centuries. T. pallidum is microaerophilic. It utilizes glycolysis for the production of its energy. This bacteria multiplies via binary transverse fission. It lacks LPS, but it has a large amount of lipoproteins, which results in the inflammatory response in the human body. This pathogen is only spread via human reservoirs, and it uses invasive mechanisms to survive in the host body. T. pallidum is closely related to other Treponema species, but they differ in transmission pathways as well as the presentation of the diseases. Syphilis has an incubation period of 10 to 90 days. When contracted, syphilis has 3 stages. There's the primary stage, which includes genital lesions, the secondary stage, where the symptoms are widespread lesions across the body, and the tertiary stage, which results in cardiovascular and neurological issues. T. pallidum can coexist with other STIs like gonorrhea. It was previously thought to be related to gonorrhea because the two diseases were often concurrent within the host's soft tissue, and bone lesions can happen in this advanced untreated state. Tertiary symptoms often include dementia, strokes, blindness, and the loss of proprioception. Congenital syphilis often results in fetal death or birth defects. Syphilis almost reached eradication, but unfortunately has had a resurgence in recent decades. The discovery of penicillin has been the cornerstone of syphilotherapy. Since penicillin was not discovered until the 20th century, tertiary cases of syphilis were historically more common, and there are speculations on which historical figures had the disease. In some cases, syphilis can spontaneously resolve during the earlier stages, and 30% of untreated patients go on to develop tertiary syphilis. Syphilis became prevalent during the 16th century in Renaissance Europe. It is stipulated that the disease was brought to Europe from the New World, with Christopher Columbus's arrival in 1493. However, there are a large number of hypotheses, and these are divided into Columbian and pre-Columbian frameworks. There's no real way to determine which is accurate or true in terms of the origin. Lesions due to syphilis can occur on human bones, and because of this, paleopathologists and osteologists have worked to posthumously diagnose historical figures with syphilis. Experts of trypanematosis make educated stipulations using biological advancements as well as analyses of historical texts and artworks. Neurosyphilis, which can develop during the tertiary stage, has had a profound but often overlooked impact on history. It can lead to paranoia, erratic behavior, or even cognitive decline. Imagine men in positions of power or renown throughout history and how this disease could influence critical decisions that shaped the history of nations. Historians have speculated that untreated syphilis may have affected the mental stability of figures like Ivan the Terrible, King Henry VIII, the Prince of Moscow, and even Nietzsche. Everyone except Nietzsche here are stipulations. The ripple effects of the disease can be vast and serves as an example of how bacteria can silently alter the trajectory of the world. Nietzsche suffered a psychotic break during the end of his life, which was attributed to neurosyphilis. He's a good example of what neurosyphilis, how it can affect someone in advanced stages, as one of the greatest minds of his era was reduced to madness. Syphilis has shaped the world through individuals as well as its broader medical and social impacts. Even today, syphilis remains a global health concern, with rising infection rates due to the lack of healthcare access and sexual education. As a disease, syphilis has acted as both a medical challenge and a social mirror.
Today we're exploring the fascinating world of Vibrio, a genus of bacteria with a significant presence in aquatic environments. These microorganisms are not only crucial ecological players, but also include species that can profoundly impact human health. Vibrio species are primarily found in water, thriving in conditions ranging from salty seas to brackish estuaries. Their roles are remarkably diverse, from engaging in beneficial symbiosis to causing serious diseases like cholera. Vibrio bacteria are typically recognized by their gram-negative cell walls and their characteristic curved rod morphology, although some may appear as straight rods. A key feature for many is a single polar flagellum, a whip-like appendage that propels them through aquatic environments, enabling motility. As gram-negative organisms, their cell envelope includes an outer membrane containing lipopolysaccharide, or LPS, which can function as an endotoxin. In the broader classification of life, Vibrio belong to the domain Bacteria. They are part of the phylum Proteobacteria, specifically falling within the class Gamma Proteobacteria. This large and diverse class includes many other well-known gram-negative bacteria. Within this framework, the genus Vibrio contains significant species such as Vibrio cholerae, the causative agent of cholera. Vibrio parahemolyticus, often implicated in seafood-borne gastroenteritis. And Vibrio vulnificus, known for causing severe wound infections. Vibrio species are chemoheterotrophs, meaning they derive both their energy and carbon from organic compounds. They are generally facultative anaerobes. This gives them metabolic flexibility. They can perform aerobic respiration if oxygen is present, which is more energy efficient. But they can also switch to anaerobic respiration using alternative electron acceptors or to fermentation pathways when oxygen is absent. A common biochemical characteristic used in their identification is being oxidase positive. Regarding their nutritional needs, a notable trait of most Vibrio species is halophilicity. They require sodium ions for growth, which aligns with their prevalence in marine environments. Under optimal conditions such as suitable temperatures, salinity, and nutrient availability, Vibrio are known for their rapid growth rates. They reproduce asexually primarily through binary fission, where a single cell divides into two genetically identical daughter cells, allowing for a quick population expansion when conditions are favorable. In their natural habitats, Vibrio species don't exist in isolation. They interact extensively with other microorganisms. These interactions include competition for available nutrients in space. Many Vibrio are adept at forming biofilms, structured communities of cells encased in a self-produced matrix, which can enhance their survival and persistence. Furthermore, they actively participate in horizontal gene transfer, exchanging genetic material with other bacteria, which can lead to the acquisition of new traits, including antibiotic resistance. Vibrio's interactions extend to larger organisms and their physical environment. They commonly associate with marine animals, notably shellfish, where they can be found on surfaces or within tissues. A classic example of a beneficial interaction is the symbiosis between Vibrio fiskeri and the bobtail squid. The bacteria colonize a specialized light organ in the squid and produce bioluminescence, which the squid uses for camouflage. With significant threats, Vibrio cholerae, with its potent cholera toxin, causes severe diarrheal disease. Vibrio parahymolyticus is a common agent of foodborne illness from contaminated seafood. Vibrio vulnificus is particularly dangerous, capable of causing severe wound infections from contaminated water exposure or life-threatening septicemia if ingested, especially in immunocompromised individuals. Research such as the genomic studies by Heidelberg and colleagues on V. cholerae continues to shed light on their pathogenic mechanisms. In conclusion, the genus Vibrio comprises a diverse and adaptable group of aquatic bacteria. They exhibit unique structural and metabolic features, engage in complex interactions within their ecosystems, and have profound impacts ranging from beneficial symbiosis to causing significant human diseases. Their sensitivity to environmental factors also positions them as important indicators of aquatic ecosystem health in a changing climate. So the next time you dive into sea salt waters, please make sure you don't have any cuts or wounds. And the next time you want to indulge into some seafood, please make sure they are thoroughly cooked to prevent yourself from getting infected from the flesh-eating bacteria.
Hidden in the soil and in sealed cans, while at the same time boldly redefining the cosmetic industry, Clostridium botulinum is a microbial paradox. There are a few other bacteria that at the same time invoke fear and fascination quite like this one. While its name is associated with a deadly foodborne illness, it also represents one of the most important substances at the intersection of aesthetics and medicine, Botox. This duality of destruction and beauty makes C. botulinum a remarkable subject of microbiological inquiry. C. botulinum is a gram-positive, rod-shaped bacteria characterized by its hardy spore-forming structure. Its shape is typically straight or slightly curved rods with rounded ends. A defining anatomical feature is its ability to form endospores, which enables it to survive extreme environmental conditions, including heat, desiccation, and a lack of nutrients. When exposed to anaerobic environments and the right temperatures, these spores can germinate into vegetative cells that release botulinum neurotoxin, or BONT. This capability explains why improperly canned or vacuum-packed foods are at a heightened risk for botulism, and care must be taken to ensure proper processing of these products. C. botulinum is also an obligate anaerobe and is unable to survive in environments with oxygen. It derives energy through fermentation pathways, metabolizing amino acids and carbohydrates into organic acids, alcohols, and gases. Its production of botulism neurotoxin is typically associated with the transition from exponential growth to its stationary phase. Environmental stressors such as pH, temperature, and nutrient availability have a significant influence on toxic production. Ingestion of food contaminated with BONT-producing C. botulinum can lead to botulism, which is a rare but potentially fatal disease. The associated symptoms are muscle paralysis, respiratory failure, and cranial nerve impairment. Symptoms typically begin within 12 to 36 hours after exposure and progress rapidly without medical intervention, which means that it's critical for patients experiencing these symptoms to seek care right away. Infant botulism occurs when spores germinate in the immature gut of infants, leading to toxin production. Although it is less common, wound botulism occurs when spores contaminate open wounds. This form of botulism is especially a concern among intravenous drug users. Iatrogenic botulism is a newer concern and has emerged from improper administration of therapeutic or cosmetic BONT doses. Animal botulism is a form that's especially prevalent in cattle and waterfowl and is responsible for significant agricultural and ecological losses and is linked to anaerobic decomposition in stagnant environments. The environmental and pathological impact of C. botulinum is extensive and should not be underestimated. Once its spores are released, they can contaminate the soil as well as aquatic systems for decades, making remediation efforts extremely difficult. As a result, public health systems prioritize surveillance and response protocols due to the potential for large-scale outbreaks, especially in emergency or low-resource settings where food safety protocols might be compromised. Additionally, due to its potency, BONT is classified as a Category A bioterrorism agent by the CDC. Even just a few nanograms of the toxin can be fatal, which goes to show the importance of rigorous biosafety practices in research and pharmaceutical manufacturing. Outbreaks require rapid detection, antitoxin distribution, and supportive care to minimize morbidity and mortality, which places significant strain on health systems when they occur. Despite being notoriously deadly, C. botulinum has been transformed into a powerful form of therapy through the purification and controlled application of BONT, which is marketed as Botox and Dysport. When injected in tiny, medically regulated doses, BONT inhibits the release of acetylcholine at neuromuscular junctions, which effectively paralyzes targeted muscles. It was initially developed to treat crossed eyes and eyelid spasms. However, Botox is now used to treat chronic migraines, cervical dystonia, spasticity, overactive bladder, and hyperhidrosis, among other conditions. In cosmetic dermatology, it is widely used to reduce facial wrinkles and signs of aging, which generates billions of dollars in global revenue each year. Furthermore, researchers continue to investigate new therapeutic applications of BONT, including its impact on depression, atrial fibrillation, and gastrointestinal disorders. Its high specificity and long-lasting effect at neuromuscular junctions makes it unique for its potential in targeted drug delivery and neuromodulation therapies. As we've discussed, C. botulinum is a microbe of paradoxes. It comes from an ancient phylogeny, and yet it is medically and culturally relevant. It has the potential to cause death and destruction, and yet its capacity to heal and improve lives has been proven and will continue to be explored. Its spore-forming anatomy, toxicity, and complex lineage reveal a microorganism that defies easy classification. Its unique characteristics make it a critical player in both pathology and biotechnology. Perhaps most fascinating, its legacy spans from biological warfare to beauty clinics, showing us that even the most dangerous organisms can be harnessed for good. Continued research on C. botulinum not only protects public health, but also exemplifies the potential of microbiology to uncover powerful tools in the most unlikely of places. I wanted to include a picture of my art project as well. I used pasta to make a portrait of a C. botulinum cell. So you can see on the outside it has flagella, pili, and a gram-positive thick peptidoglycan cell wall. And then on the inside there's a cytoplasmic membrane, an endospore down at the bottom, the nucleoids, some ribosomes, some plasmids, and then some of the BONT toxin up at the top there. Thanks for listening.
Helicobacter pylori, the stomach invader, written and illustrated by Melinda Powell, with an artistic, artful contribution by Simone. Mission arrival. Meet Helicobacter pylori, aka H. pylori, a spiral-shaped, coiled-structured bacterium built to survive one of the most hostile environments, the stomach. H. pylori is a gram-negative bacteria with 5 to 11 flagella to move and burrow in the stomach lining. It's less than 5 micrometers in diameter. H. pylori has a history of following the migration patterns of humans and have been infecting them for close to 100,000 years. They are highly diverse in their genome because of mutations, recombinations, and transformations. The establishment. With the help of urease, H. pylori neutralizes stomach acid into ammonia and carbon dioxide, creating a toxic bubble, allowing the bacterium to slip beneath the mucus layer. Because H. pylori can thrive in low oxygen levels and take in glucose for energy and carbon, but can also use amino acids and lipids, H. pylori can break down molecules using aerobic and anaerobic metabolic options. It reproduces primarily through binary fission and, to grow in the lab, multiplies best in a complex medium with blood or serum. Chaos ensues. H. pylori changes the pH and oxygen levels in the stomach, causing a disruption of the microbiome and a mass exit of the typical healthy microbes. The disturbance often triggers chronic inflammation or gastritis. Some of the affected decreasing microorganisms are Actinomyces, bacteroids, and Clostridium. A couple of the increasing microorganisms are Proteobacteria and spirochetes. The ulcer is born. H. pylori weakens the protective mucus layer of the stomach, and the acid present is able to erode away the lining, creating peptic ulcers. Ulcers are painful. They can be in the stomach and duodenum. They can also be caused by long-term use of certain medications. Ulcers become more complicated if left untreated, leading to bleeding and holes in the stomach lining. When trouble grows, in rare cases, long-term infection can lead to gastric cancer, one of the most deadliest forms. It is important to remember that less than 3% of people with H. pylori actually develop stomach cancer. Mucus-associated lymphoid tissue, also known as MALT, lymphomas of the stomach, are also thought to be linked to H. pylori infection. Science strikes back. Science has weapons. A combination of antibiotics and acid blockers are taken to clear the infection and heal the stomach. Beware, antibiotic resistance is a concern due to horizontal gene transfer, so more than one antibiotic is usually recommended. If you or someone you know has questions about H. pylori, you are not alone. Contact your nearest microbiology class, microbiologist, or doctor's office for support. The end.
Hi there, I am Yersinia pestis. I'm a gram-negative zoonotic pathogen. That means I transmit from animals to humans. I have killed millions in the 14th century. I transfer to humans through the bites of infected fleas that live on rats. I form in Coccobacillus formations with my friends. I am non-motile and non-spore forming. I am surrounded by a thin peptidoglycan layer. Antibiotics like dioxycycline is now the first line of defense for Y. pestis and could have saved millions of people from their horrible death in the past. I developed 6,000 years ago and continue to acquire my virulence from horizontal gene transfer. In the stomach of a flea, I form a biofilm that blocks proventricles, causing regurgitation and transmission to the new post. As a facultative anaerobe, I can survive with or without oxygen using glycolysis and the pentose phosphate pathway. I prefer to live at 28 to 30 degrees Celsius, but I can survive at the human body temperature too, perfect for living in fleas. Humans act as incidental hosts, rodents as reservoirs, and fleas as vectors. Hey, look, a free ride. I am known for my pathogenicity. However, recently it was discovered that I have a symbiotic relationship with lytic bacteriophages, which plays a role in my evolutionary fitness. With how contagious I am, I can easily spread through the town via contact with contaminated tissue or flea bites. I can live inside amoeba during interepisodic periods, specifically Dictyostelium disodium, that makes amoeba a potential plague reservoir. Because I can live in soil and water for at least 24 days, I have an upper hand on persistence and transmission. It is unsure how climate change will affect me. Thank you for listening to my story. Until next time.